Here we are looking at microbial phylogeny, and phylogeny is a word you don't hear very much outside of biology. It's the study of evolutionary relationships. So if you think about how we are related to chimps, or dogs are related to other dogs, or you're wondering how is a cat related to a tiger, you're thinking about phylogeny. And naming them, classifying them is taxonomy, but we typically want that to be based on phylogeny. So we want to think that two separate species share an evolutionary history that branched at some point and they became independent at some point. But before that point, they had a common ancestor. And that's really how we look at all of life on Earth. We kind of look at common ancestors. But there are only a few things I want you to learn about phylogeny for this course, and we'll get into them. So this slide says what I was just saying. Taxonomy is systematic classification, and phylogeny is evolutionary relationships. So what you'll find on this slide, if you want to go back and look at it, is statements you could make about taxonomy, and then statements you could make that show phylogeny. So this is what you'd say if you were doing taxonomy. This is what you'd say if you're doing phylogeny. Go ahead and pause the video, read through those, make sure it makes sense to you. Let's move on. So in microbiology, we want taxonomy to be based on phylogeny. So we want classifications to be based on evolutionary relationships. Two similar bacteria, we want to assume that their common ancestor was pretty recent, that they only recently became two separate species. Two very different bacteria, we want to assume that they've been separate for a long time. So the thing is, we don't always trust that, and that what do you call it? That assumption isn't always valid. And anyway, taxonomy and phylogeny are always changing because we find better and better ways to figure out the evolutionary history of organisms. We get better technology. The best example of this is DNA sequencing let us really get a much better idea of which organisms are closely related to which other ones. And then more and more clever ways of analyzing DNA sequences have led us to reclassify organisms. That's all off to the side of what we're really thinking about in this course. But phylogenies have always been based on comparisons. And when you're piecing together which vertebrate animal is closely related to which other one, you can compare the relative lengths of bones, and do they have bones, and which way do the bones point, and things like that. Of course, microorganisms don't have bones, and under the microscope, they all look pretty much the same. So it has always been hard to figure out their phylogeny, and for a long time, what comparisons did we do? We did biochemical tests. We saw what enzymes do they have? What chemical reactions do they have? We did that because that was easy to do. Now we use DNA, and now we've realized that sometimes phylogeny is difficult or even impossible to understand because several reasons. One of them is horizontal gene transfer. That means genes from one species can actually hop into another species, and that just ignores their evolutionary history completely. That can happen today and not back in history when those species split. So that kind of scrambles the DNA a little bit and makes some phylogeny truly unknowable. And this is what I was saying before. As we get new technologies and new ideas, phylogenies get updated. Okay, cool. So the biggest advance in phylogeny in the last several hundred years was the idea of sequencing ribosomal RNA. If you go back and look at what is a ribosome, what is its structure, you'll see that an essential part of a ribosome is its own structural RNA. So pause the video and remind yourself, what is a ribosome? And then go back and look, and what you'll see is a ribosome is made of proteins, and it's also made of RNA molecules that just act as structures that hold the proteins together. It's wild. We call that ribosomal RNA, and that RNA is never translated. That's not its job. Its job is to be part of a ribosome. But that RNA is encoded by genes. It is normal RNA. So it turns out that that's a good example of sequences to compare between two organisms. So doing that, we get this phylogeny, this universal phylogeny of all life on Earth. So if we compare ribosomal RNA genes, so the genes that encode that RNA in those ribosomes, this is what we get. We see us over here, the animals, and the fungi and the plants near us. We see over here the bacteria all together. And then we see something called archaea in the middle. If we were to look at all of these things under the microscope, the bacteria and the archaea would be the prokaryotes, and eukarya would be the eukaryotes. So all of these would have nuclei, things like that, and all of these would not. These would all be small and more simple structurally. So what I want you to do in this phylogeny section is learn how to read a tree like this. Learn how to interpret it and see what information it gives you, what information it doesn't give you. So the way you use it is you pick two organisms, like say animals and say flagellates, and you measure the distance between them, the shortest possible path. So I'll trace the shortest possible path. You'd go down here to this junction, and then follow it to this junction, and then to this junction. So there you will have measured a path between two different organisms. So, and what that path relates to is how many differences there are between their 16S ribosomal RNA genes. And that, we assume, could be a proxy for how many differences are there in their genomes, and how different are they, and how evolutionarily distinct are they. So that's what we get. So this is how we read the universal phylogeny, and here's some examples. Animals and fungi, how different are we from the fungi? Well, we would think, I'm not that similar to a mushroom, I'm not that similar to mold. And so we can measure this distance, and on this phylogenetic tree, that distance isn't very big. And that's because there are organisms out there that are much more distantly related from each other than those. So within the bacteria, two different phyla of bacteria, Flavobacteria and gram-positive bacteria, are much, much more different from each other than we are from fungi. Even the best example of this is within this phylum Proteobacteria, two closely related species, Escherichia coli and Salmonella and Enterica. They look the same in many ways, but they are more different from each other than we are from dogs. So that's really what you do with this tree, is you say, are these ones more closely related to each other or more distantly related than these two are from each other? You can do some other things, like it tries to show us a little bit of evolutionary history. So where these branches meet kind of corresponds to how far back in time the common ancestor was. So it's like as you go forward in time, there was an event where the diplomonads split off and became different from everything else. There was an event where entamoebae split off and became different from everything else. And there was an event where the animals and fungi split off from each other and became different. So as you go back in time, you're seeing more and more primitive ancient events. And way back in time, you see when the archaea split off from the eukarya. And the archaea are a lot of weird organisms. All the way back in time, the bacteria split off from everything else. So even though they look the same under the microscope and they're both prokaryotes, bacteria and archaea are only very distantly related. If you choose like something like methanopyrus and compare it to any bacterium, the path between them is very long. They're very different from each other. So that's cool. So what I want you to do with this is look around and just get used to this big structure with these 3 big domains of life. These are the big domains. This is eukarya, archaea, and bacteria. And then, so spend some time with that, see if there's anything on there you recognize. Like this thing here you might recognize, this thing here you might recognize. And then pause the video to do that, and then move on to the next, we'll move on to the next slide. So phylogenies aren't always perfect, and we keep seeing this weird disagreement where, depending on how you analyze the DNA, you get 2 different, very different base level trees. So this 3 domains tree, this is a simplified version of this tree, which has 3 domains. It has the bacteria over here, and then the archaea and eukarya split from each other. And it has 3 different big groups of archaea. And so that's what we see here. The bacteria split off first, then the eukaryotes split off, and then the big archaeal groups split off from each other. And this is what you get if you compare ribosomal RNA sequences to each other. This is the tree you get. This is the evolutionary relationships you infer from that. If you use other genes, you sometimes get this other completely different result where the bacteria split off first, but then the archaea, an archaeal group, split off, and then later 2 archaeal groups split off from the eukaryotes. In other words, there are 2 domains in this tree, and we really belong in the archaea. So the eukaryotes, cells with nuclei, animals, plants, fungi, all of us would be in the archaea. Whereas the 3 domains tree show bacteria over here, very different from everything else, the archaea as one group, and then the eukaryotes as another group. And no one knows which is right. So there's that. So I might also show you this tree, which is the same thing. This is the 3 domains ribosomal RNA tree. It's just been graphically simplified a little bit. And this shows the hypothetical movement of cyanobacteria into some eukaryotes. And it kind of shows you when in evolutionary history photosynthetic plants ate some cyanobacteria, which became chloroplasts. So these would be chloroplasts. And at some point, a member of the proteobacteria got eaten and became a mitochondria. And that was much more ancient. So this is kind of cool, because what we could see is that these diplomonads, for example, don't have chloroplasts. So, and the parabacillids don't have chloroplasts. So chloroplasts came over sometime after those split off. That's the kind of thing you can do with phylogeny. And I just want you to recognize this tree, recognize how close together the animals, fungi, and plants are. Whether or not there are truly 3 domains or 2 domains, either way, the multicelled organisms are pretty close together on this phylogeny, and the bacteria are super far away. Practice drawing a path between two different organisms. Practice drawing that shortest path, because that's really what you have to do to read this kind of tree. You don't look at how close the amoebas are to these things over here. You look at how close they are this way. Always measuring the path. All right, enough of that. Let's move on to the next video. If you are watching this in winter of 2023, this is the last video for Unit 2. And if you're watching this in a different term, you might want to talk to the professor about whether there should be more videos or not. I'll talk to you later.